Wish you a very good morning to all. So, we are in our biology and I am going to start last and final chapter of your syllabus, that is chapter number 2, sexual reproduction in flowering plants. So, here one PPT PowerPoint presentation for you, and you will be studying in detail how the sexual reproduction will be occur in the flowering plants. So, as all we have been taught in our previous classes, the reproduction in plants, in that the type of reproduction we discussed, in that sexual reproduction also we discussed, asexual reproduction also we have discussed. So here some typical flower we are taking. If you know that in 6th standard, 7th standard, even in 8th standard, there was one topic of typical flower. Draw a level diagram of typical flower. In that we were discussing about which is a male reproductive part of the flower, which is a female reproductive part of the flower. So in that, now in this we are studying all the procedures, all the stages in the sexual reproduction of the flowering plants. So here you will see that the sexual reproduction in flowering plants, it is a one way or another way. So all the flowering plants generally we call as a angiosperms show a sexual reproduction, and flower are the site for the sexual reproduction. So only one part of the flower, one part of the plant that is responsible for the reproduction process, so that is the flowers. So flowers are main and special part which is responsible for the sexual reproduction. So here different slides you will see the several hormonal and structural changes result in the differentiation and development of the floral primordium. Okay, floral primordium, how the flowers are changed, okay, from typical to the main growth. So you will see here one photo there, so in that also you will understand. And the fluorescence, here the floral buds and then the flowers. So how the flower is totally germinate. Okay, the germination process you know, and in that the floral buds and then it becomes flower. Just example take of this lotus. So first it was in a bundle. When it will open, you will see the different stages in the flower. Okay, so that all things we need to discuss. So here the structure of the flower, and we have drawn this type of diagrams, and this may be asked in the exam. Draw a level diagram of stamen and stigma. If you know that the main carpels, that is gymnosiums and androsiums. Gymnosium and androsium. Gymnosium we are using for the carpels, or we can use for the stigma. Okay, and the androsium we are generally use this word for that stamens. Stamens are the male reproductive part of the flower, and the carpels or stigma is a female reproductive part of the flower, that you are aware. So here, when you see this diagram, you will remember your previous years. When you remember your previous year, you will understand that the typical flower already we have drawn, and I was taking your class, if you know that. So in that I was teaching you, and I have drawn this type of diagrams on the board also, and it was asked in the exam. And here the typical flower has two parts. One is an androsium and second is a gynosium. Androsium and gynosium. Androsium for the male. Remember that androsium procedure for the male and gynosium procedure for the female reproductive part. So that you are aware. So here the structure of the flower. So in that androsium when we talk about, so it is the male reproductive part of the flower, and it consists of whole of stamens, and their numbers and strength are variable in the different species. In different variety of plants, their stamens numbers are different. Okay, so that also we need to take care. So here you will see the this is the style. So this part is the filament, and here these are generally we call as a stamens, or generally it is anther. This part is anther. So here you will see this part, the connecting inside some stamens are there. Inside the anther, generally it is called the pollen grains. Pollen grains you notice. Okay. So floral axis are there. Then articulation, and then pedicle. So that is providing the base of the flower, and nectary which is connected to the ovary. Okay. So that you know. So parts of the stamen. The parts of the stamen in that filament and anther. Filament and anther. Here you will see. This is a filament. This is the anther, and inside anther stamens are there. Okay. So generally we call the pollen grains. And perianal, that is petal, that is corolla, sepal, that is calyx, and all the flowering parts you will see here. Okay. So two main parts of the stamen, that is filament and anther. The parts of the stamen, first we will discuss the filament. Filament is stalk, which providing one support. So filament is a long and slender stalk, and is proximal and is attached to the thalamus or the petal of the flower. So here you can see this part. Okay, it is attached to the petal and the thalamus. So here it is a main function of the filament is to provide strength, provide long ends and the stalk, which is providing the support to the anther. Okay. Next is the anther. Where is the anther? Anther is here. You can see here. So terminal and typically bilobe. It is bilobe. You can see structurally also you can see bilobe, and each lobe has two thecae. That is called dithecus. Okay, and often a long longitudinal groove run lengthwise separating the theca. So it divides into two parts. It divides two parts. One part here. Suppose here you can take example this two. So it divides one part and the second part. So it makes two theca. Next, the process of the androsium. So here the anther is a tetragonal structure consisting of 4 microsporangia located at the corners, 2 in each lobes. So here you can see here the 4 lobes are there, and inside pollen grains are there. So these microsporangia develop the pollen sacs, and they extend longitudinally all through the length of an anther and are packed with the pollen grains. So inside the pollen grains will grow. So these pollen grains, these are the sacs, and inside the microspores, mother cells we can say, and tapetum. Okay, so that you need to understand. So a structure of the microsporangium. The structure of the microsporangium already there in the textbook. So anther sac with the pollen filament. So this you can see the pointed part. You can see the pollen filaments. This is a microscopic structure. This is the section, and you will see that the microspores, mother cells, and middle layers and androsium, all things you will see. So here, microsporangium. A typical microsporangium is near circular in outline. It is a circular, and it is surrounded by 4 wall layers. One is the epidermis, which is outermost, endothecium, then middle layers, and tapetum. So here you can see the tapetum also, microspores, middle layers also, endothecium, and epidermis. Epidermis which provide the outer support, and the outer 3 layers give protection and help in the sense of anther to release the pollens. So when the pollen grains are mature, that time it will release them. So before that, these 3 layers are protecting them: endodermis, structure of the microbe, you can see the tectum. The tectum is here. You can see this part. I am talking about this part. The tectum is the innermost layer. Nourishes the developing pollen grains, which providing the nutrients, providing the nourishment. And cells of the tectum contains a dense cytoplasm and generally have more than one nucleus. So it is only nucleus, bipolar nucleus, we can say more than one nucleus. And in young anther, each microsporangium has sporangious tissue at the center, and it consists of compactly arranged homogeneous diploid cells. Diploid cells means two, and the sporangious cells. Okay, so inside also, if you see the structure, they are sporangious tissues, and they are diploid. So they generally we call as the diploid cells and the sporangious cells. So androzium, it is a microsporanginesis. The process is called microsporanginesis. So as the anther develops, each sporangineous cells, that is microscope or pollen mother cell, undergoes mitotic divisions. You might have taught by Sahil sir what is mitotic division and what are the processes, what are the steps involved in that to perform a microspore tract. That is, the microspores arranged in a cluster of 4 cells. So here you can see the clusters of 4 cells, 1, 2, 3 and 4. 4 cells are arranged, and the formation of microspore from the pollen mother cell, that is a PMC. Okay, Prime Minister, it is not like that. PMC, Prime Minister Chandu. Remember that Prime Minister Chandu. It is not Prime Minister Chandu. It is pollen mother cell. PMC for the flower. Got it? And that whole process, meiosis, is called the microsporanginesis. Microsporanginesis. What is microsporanginesis? So it is the formation of microspores from a pollen mother cell, PMC, through a meiosis process is called microsporanginesis. Got it? So definition you need to remember. And here also, as the anthers mature and dehydrate, the microspores dissociate from each other and develop into pollen grains. Now, how the pollen grains develop? This is a procedure. Each microsporangium contains thousands of pollen grains. They are released with the dehiscence of anther. Means how they release from 3 layers. So that also, when it gets burst means open, and the pollen grains comes out. Okay, so that it's a procedure. Now pollen grains, they are the main gametophytes. As I said, the pollen grains, if you see the structure of pollen grains, generating cells are there, micronucleus, cytoplasm there, enzyme there, then vegetative cells are there, germ spore, and thickening of the cellulose. Now cellulose, which is available only in the plants that you know, and generally it is a spherical. You can see it is a spherical, 25-50 millimeter in diameter, and the cytoplasm is surrounded by the plasma membrane. It's called plasma membrane, and the pollen grains has 2 layers, that is exine and antene. So exine, which is outside, antene, which is somewhat inside. Okay, exine and antene. So 2 layers are there. So that first, what is exine? What is the role of exine? So here the hard outer layer made up of sporopollenin, and it is a highly resistant organic material. It can withstand high temperature and strong acids and alkali. So at any of the temperature, this will be providing the full support to the pollen grains, and enzymes cannot degrade sporopollenin. So enzymes, any enzymes will be there, they are unable to degrade them. And the exine has a apertures called germ spore. You can see when they will come out. sporopollen is absent. So here sporopollen is not observed, and pollen grains are preserved as fossils due to the presence of sporopollens. Here fossils, pollen grains are preserved as fossils. Fossils, you know that it's a dead material which will be prevented for a long time. Fossils, you are aware, and for exhibit patterns and designs. So different patterns of flower you will see. Different types. Suppose we will take example of rose, lotus, sunflower. Different patterns are there that will be decided by the exhibit. The main function of the exhibit is to exhibit the patterns and design of the flower. Remember? Okay. Main function of the exhibit is to exhibit patterns and design of the flower. So 18. So 18 is the inner wall, which is out inside the exit, and it is a thin and continuous layer made up of cellulose and pectin. The two main proteins are there, cellulose and pectin. Okay, so in exam you may be asked, the antin is a thin and continuous layer made up of A, B, C, D. So it is cellulose and pectin, cell wall and pectin, then sucrose and pectin. It may be anything. It may ask any form, but the answer is cellulose and pectin. Next, that is pollen grains. Mature pollen grains contain two cells that we know. That is vegetative cells and generative cells. Two cells, vegetative cells and generative cells. Now, what is the role of vegetative cells and generative cells? So here vegetative cell, it is a bigger, has abundant food reserve and a large irregularly shaped nucleus. So in that, the nucleus is not in proper shape. And generative cells, it is a small and floats in the cytoplasm of the vegetative cell. It is a spindle shape with a dense cytoplasm and a nucleus. So that the biggest difference between vegetative cells and generative cells. So here vegetative cells are bigger, generative cells are smaller. Here vegetative cells irregular nucleus and generative cells have regular nucleus with a dense cytoplasm. Okay, you can see that. In the structure also you can see. So over 60% angiosperms shed their pollen grains at 2-cell stage, and in others the generative cells divides mitotically to give 2 male gametes, and thus pollen grains are shed at 3-cell stage. So 2 male gametes and it sedded at 3-cell stage. So that you have to see. So here some pictures are there. Structure of the flower and rosette. The seeded pollen grains have to land on the stigma before they lost viability. Now this procedure, you know that pollination process, how the pollen grains transfer to the stigma, pollination process. Okay, that you know. And the viability period of the pollen grains is variable. It depends on the temperature and humidity. At what time the pollination process will be carried out, that the factors are responsible. And the pollen grains, when it will transfer, that time also condition also useful. So viability of pollen grains of some cereals, rice, wheat, etc. is 30 minutes, and some members of leguminaceae, rosy and solanaceae have variability for months also. So some pollen grains, when it transferred to the stigma, it can start proceed within 30 minutes, and some have more variability, so that's variability for the months also. So that the role of the pollen grains. Now economic importance of the pollen grains. Now these rich in the nutrients, okay, because they have to fertilize the ovum, and pollen tablets are used as a food supplements. Pollen tablets and syrups increase the performance of athletes by knowing just this thing, and we will have this type of pollen grains, it will be dangerous for any of the people without doing diagnosis. So must consult with the doctor and then only go for this type of medicines, this type of what we can say, syrup or tablets. Okay, so without consult doctor, don't go for it. Now, gymnasium. Gynosium, it's a female reproductive part, and it has a single pistil. It may have a single pistil, monocarpellary, or more than one pistil, that is multicarpellary. So here you can see the stigma, style, ovary, thalamus structure. The stigma which stick the pollen grains. Carpels, which is in different shape, and multicapillary pistils may be fused together. That is a same carpus or free apocarpus. Okay, that each pistil has three parts: stigma, style, ovary. This already we know. Functions of each also we know. What is the function? Stigma, landing platform for the pollen grains. It catches the pollen grains. Then the style, elongated slender part beneath the stigma, which is connected with it, and ovary, basal bulb part, which start the fertilization. So here the procedure, the whole gynosium procedure steps, you will see stigma, style, and ovary. So inside the ovary cavity, there is a locus in which the placenta is located. Here inside, this part I am talking about. And arising from the placenta are the ovules. There is a megasporangia, and the number of ovules in an ovary may be one, may be many. So if it is one, wheat, paddy, and mango example we can take. If it is many, there is multi, there is papaya, watermelon, and orchids. You might have seen that inside when you open the watermelon, the black ovary, that is ovary only. So you will see that. And the structure of the megasporangium, ovule, you can see already. We have seen ovule, funiculus, and placenta. That is ovary or locule. So here the procedure, placenta will be nurtured, and funicule that provide the full fertilization process. So ovule is attached to the placenta by a stalk, that is a funicle, and the junction between the body of ovule and funicle is called hilum. It is called hilum. You can see the word hilum. It is this part, hilum, this part. Okay, that is called hilum, and each ovule has one or two protective envelopes, that is gametes, except at the tip where the small opening microphyll is present, and opposite the microphylla end is the chalza, that is a basal part, that is providing support. So enclosed within the integuments, there is a mass of the cell called nucellus, and its cell contains reserved food material. They are providing nutrition, like pollen grains, you know that. And inside the nucellus is the embryo sac, that is a female gametophyte. Embryo, you know, and the ovule generally has a single embryo sac formed from the megaspore. So that is the whole process. Calanzal pore, that is embryo sac, nucleus inside, integument, outer integument, microphylla pore, microphyll, funicle, and hilum. Okay, so megaspore mother cell, MMC, PMC, that is pollen mother cell, MMC, that is a megaspore mother cell. Remember this too. PMC, that is pollen mother cell, MMC, that is megaspore mother cell. Remember this, okay? So here it is the formation of megaspore from megaspore mother cell. Ovule generally differentiate a single MMC in the microfilm region of the new cells. It is a large cell containing dense cytoplasm and prominent nucleus. So MMC undergoes the meiosis to produce 4 megaspores. So here meiosis process will be carried out, and embryo sac. The majority of flowering plants, 1 megaspore is functional while the other 3 degenerated. Okay, 4 are there but 1 only functional and 3 will be degraded, means degenerated. So functional megaspore develop into the female gametophyte and other 3 which are degenerated, and the embryo sac formation from the single megaspore is called monosporic development. Single will be there, so it is called monosporic. Okay, the nucleus of the functional megaspore divides mitotically to form 2 nuclei, and they move to the opposite poles forming 2 nucleated embryo. You can see that 2 to 4, 4 to 8, and then it. Okay, so you know this. That is antipodals, 2 polar nuclei, central cell. It will form A and syncytids. So this nuclei again divide to 2 times, forming 4 nucleated and 8 nucleated. Blastocyst process, and this is the stage of the embryo sac. And this division are free nuclear. Nuclear division are not followed immediately by cell formation, cell wall formation. After 8 nucleated stage, the cell walls are laid down, leading to the organization of the typical female gametophyte. And 6 to 8 nuclei are surrounded. 6 to 8 nuclei are surrounded by the cell walls and organized into cells. The remaining 2 nuclei, polar nuclei, are situated below the A apparatus and the large central cell. So these 2, I am talking about these 2, and 6 to 8 are surrounded by the cell wall. A typical mature egg embryo sac is 8 nucleated and is 7 cells. So 3 cells are grouped as micropillar and form egg apparatus. It consists of 2 synergids and 1 egg cell. So here synergids have special cellular thickenings at the micropillar tip called the filiform apparatus. Where is the filiform apparatus? In this one. Filiform apparatus. And they have to guide the pollen tubes into the synergid. So 3 cells at the calendule end are called the antipodals. So antipodals, this part antipodals, they are 3 cells at the calendule end are called antipodal. 3 cells are the calendule end. So the large central cell has 2 polar nuclei. So you can see here. Okay, so here the formation of female gametophyte embryos overall. So here you can see the calendula, that is mitotic tetrad, into the 4 stages: FG1, FG2, FG4, FG5. Okay, so in that functional megaspore, at the end it will be mature female gametophyte, that is F6 or 7, and it will form 1 microfilm, that is pollen tube nerves. Alright, so this is end of course calendar, C and C, central cell. So this is about the androsium and menosium. So the structure of the flower you can see. You just go through the slides one by one and you will try to understand. Again read the textbook, all the details, study I am giving you, all the detailed information I am giving you about these 2 topics, that is androsium and menosium. So thank you, have a nice day. Remaining topic we will discuss in our coming video presentation. So thank you once again. Please go through it, try to understand.